Fluorescence Resonance Energy Transfer, FRET, by Ayodele Ogunsede. Background on FRET. Fluorescence resonance energy transfer was first described by Theodore Forester in 1946 when he presented the opportunity of retrieving information on molecular proximity on a nanoscale. The principle of FRET relies on transfer of excitation energy of a donor fluorophore to a nearby acceptor fluorophore in a non-radioactive fashion through long-range diploid-diploid interaction. FRET stands for Forester Resonance Energy Transfer. This was named after the German scientist Theodor Forester, but the term fluorescence is often used instead in scientific settings. Application of technique. Fluorescence resonance energy transfer coupled with GFP can be used to study molecular interactions inside living cells. FRET biosensors provide a means for visualizing signaling molecules in live cells with high spectrotemporal resolution. Here I have a schematic diagram of assays based on FRET. When two light-responsive proteins are very close, energy of the donor is transferred to excite the acceptor by FRET. In diagram A, protein ashtrays were set up using GFP and YFP that are tagged to proteins A and B. Interactions between A and B can be detected by the emission color change. B, we have FRET-based calcium sensors. FRET occurs only when calcium-sensitive linker changes conformation by binding and bringing two light-responsive proteins very close. This was a research I found online, experimental validation of predicted cancer gene using FRET. The researchers wanted to find a means of validating the result by MATLINK. MATLINK is a gene prioritization method that, when provided with a set of genes known to be involved in a phenotype, for example, cancer, it to produce a ranked list of genes and or proteins tightly associated with the input. In this case, the researcher they just wanted to find a way to validate using FRET to validate the result by MATLINK, so they can focus more and I guess proceed with their experimental research in confidence. So FRET was used in this experiment as a validation tool, basically. One of the query cancer genes that was predicted by MATLINK was a gene called JAK2. Through some filters that was used and other parameters that was set up, MATLINK came out with JAK2 as a protein that, as a gene that was most promising, that was the most promising candidate. And basically what they did here, they used FRET to validate the results of MATLINK of this gene, or to study the interaction of this gene in more detail. JAK2 is a non-receptor tyrosine kinase implicated in eosinophilic lymphoblastic leukemia. FRET was used to validate JAK2 and to verify protein-protein interactions with the cancer gene predicted by MathLink to focus more experimental effort on the most promising candidates. The experimental results they got after performing FRAIT, I think they found 18 other protein interactions to JAK2. Strengths and drawbacks of the experiment. It is a cost-effective experiment, and it's not time-consuming. The use of FRAIT as a validation tool provides confidence in the use of MathLink as a prioritization system of targets in the battle with polygenic diseases. Negative drawbacks. There was no negative control to prevent against stoichiometry. To prevent against stoichiometry. The phase signal may be masked by background of fluorescent labels that are not undergoing FRAIT. One of the questions I have related to this topic, if there are multiple proteins, or in this case, there were multiple proteins, but if FRAIT is used as a single molecule protein-protein interaction, so how were they able to come up with 18 other proteins that were interacting with this gene? And these are my references. Thank you.